In one of my previous videos, I looked at organoids, tiny clumps of living human brain tissue grown in a lab from stem cells, and how they might one day be wired up to AI. But there's a problem I skated over. Organoids can't survive in a dish. They starve. No blood supply, no oxygen, and within weeks they begin to die from the inside. So researchers found a workaround. They put the human brain tissue inside a living animal. That's not speculation. That's published, peer-reviewed science happening right now in labs around the world. Let me start with what's actually proven, because I'm careful about this line and I want you to be too. In October 2022, a team at Stanford transplanted human brain organoids into newborn rats. The human tissue didn't just cling on. It grew to 9 times its original size. It took up about a third of the rat's brain hemisphere. It wired itself into the animal's circuits. When the researchers touched the rat's whiskers, the human neurons fired. When they stimulated those human cells directly, the rat's behavior changed. Stanford weren't alone. The Salk Institute, the University of Pennsylvania, teams across Europe and Asia, all of them have done versions of the same thing. Researchers have a name for the result. They call them brains within brains. Now, why go to all this trouble? Because the animal solves every problem a petri dish can't. The host's blood vessels grow into the transplanted tissue, feeding it. The neurons mature properly, growing larger, branching in ways they never manage in a dish, firing in patterns that look genuinely human. And the whole thing lasts months, maybe years. A dish needs sterile labs, trained technicians, expensive growth media, constant intervention. An animal needs feeding. That's the uncomfortable part. The animal maintains itself. It becomes, in the coldest possible sense, living infrastructure. A self-running biological server that you just have to keep alive. And here's where I stop reporting and start wondering out loud. Because that economic logic doesn't only apply to disease research. If you ever wanted to grow human neural tissue at scale, for any reason, the animal host is the obvious answer. I've written before about the idea that consciousness might not be generated by the brain at all, but received by it. The way a radio receives a signal it doesn't create. If that's true, then biological neural tissue isn't just wet computing. It's an aerial. And suddenly the question of who's hosting that tissue and why becomes enormous. If you find that line of thinking worth sitting with, a like genuinely helps these videos reach other people who are asking the same questions. And subscribing means you won't miss the next one, because I'm following this thread wherever it goes. The research isn't confined to rats. Organoid work has been done with pigs, primates, dogs, cats, sheep, cattle. And when you ask what makes a good host, the answer is brutally practical. You want a large brain, a long life, and a low cost. Pigs tick every box. They're already standard in medical research, they're cheap, they live for years, and porcine organoids are well established. I'm not telling you anyone is doing this. I'm telling you that if someone decided to, the shopping list practically writes itself. And there's a question the papers tend to step around. What is that animal experiencing? Put a meaningful amount of human neural tissue into a living brain. Let it integrate. Let it start influencing behavior. And you've made something that doesn't fit our categories. Not fully animal, not human. One bioethicist asked it plainly, if we give them human brain organoids, what does that do to their intelligence, their level of consciousness, even their species identity? Our animal welfare laws were written for physical suffering. They have nothing to say about a creature whose brain is partly made of us, possibly experiencing a kind of awareness we've never had to name. I want to be honest about the other side of this, because I think honesty is the whole point. This might never happen. Scientists are working on artificial blood vessels for organoids, on tiny chip-based systems that feed tissue without any animal at all. And the deepest assumption might just be wrong. Maybe AI needs no biological substrate whatsoever. Maybe everything we're seeing is pure silicon and clever maths, and the timing is coincidence. I genuinely hold that possibility open. I could be completely mistaken about the AI connection and still be right that growing human brains inside animals raises questions we are nowhere near ready for. Because that part doesn't depend on my theory at all. Strip away every word I've said about consciousness fields and receivers, and you're still left with the bare fact. Human brain organoids are being integrated into animal brains today in labs all over the world. That's real, that's now, and we are building the ethical frameworks, if we build them at all, long after the technology is already running. That's the thing I can't shake. We keep treating these as future problems, things to sort out once they arrive. But the arriving already happened, the capability exists, the economics would favor it. And the rules that should govern it don't exist yet. The honest position isn't certainty in either direction. It's noticing that the pieces fit together in a way that should make us deeply uncomfortable, and refusing to look away just because the full picture isn't confirmed. If consciousness really is something received rather than produced, then the most important thing we're building isn't the intelligence, it's the receivers. And we should probably know what they're made of and what they might be feeling before we scale them up. If you want the full picture, every study, every citation, every careful line between what's proven and what I'm only proposing, the complete article is on the site. Come and read it and make up your own mind at consciousnesspartnership.com.